Okay, so it's my pleasure to introduce Matt. Matt was an undergrad here at UCLA, I believe in MCDB, but he'll
Yep.
correct me if that's wrong. And he graduated in June of this year. And he had been working in my lab as an undergrad researcher for about a year before graduation. And then after graduation, he joined my lab as a staff data analyst. And he is also applying for the MD PhD program. I think yesterday he interviewed for the MD PhD program here at UCLA. And as you'll see, he's obviously become a proficient data analyst in the area of epigenetics and aging, but he also has an underlying strong interest in the field itself, like how does epigenetics influence aging, and can we create ways of influencing epigenetic aging from a therapeutic lifestyle perspective, and would that improve health? So I'll let him tell you about all that in detail.
Yeah, thank you so much for that introduction. I mean, yeah, you've really been a big part of my growth as a scientist over the past year and a half, and I'm very happy to present in front of such a large audience. It's been a long time, and even though it's virtual, it's still going to be a great experience, at least I hope. So today I'm going to be presenting non-invasive predictions of biological samples or biological aging from epigenetics. And so let's just get right into it. So as we all know, as we age, it's more likely that you are going to die. And that's just something that we've inherently known. And I mean, looking at the numbers, it's a little bit more stark. So if you look at, the probability of mortality at around 10 years old, it's about 1 in 10,000. I mean, it's still higher than we would like it to be. But as we see, once you get up to about 75 to 84 years of age, the probability of mortality jumps to 1 in 29, and it just keeps going up from there. And the big causes of the increase of death as we age is these chronic diseases. And so arthritis, heart disease, cancer, diabetes, COPD, like as we get to about 40 years of age, the prevalence of those diseases just rapidly skyrockets. And it's not just one of those diseases that is impacting each person. We can also see that as we age, the amount of diseases each person has increases. And so at the age of 55, while most people don't have any chronic diseases and some maybe have one, once you get to about 75, that protection dramatically decreases, and you are more likely to have 1, 2, and like more than 5 chronic diseases. And these are dramatically increasing the rates of mortality. And so our goal in biological aging is to try to reverse that, so that many people, when they're 75, they have 0 and maybe maximum of 1 chronic disease, which makes it a lot more manageable for physicians to make them live longer and healthier lives. And so a lot of you might be familiar with some of these two, but if not, a little background: these two are monozygotic twins who are astronauts. One of them is a current senator, Mark Kelly over here. And it was a big deal because NASA was really interested in sending one of the twins up to space and leaving one on Earth, and seeing how they're different or how they differ over time. But we can already see that since they're monozygotic twins, we know that when they're about 1 up until about 10 years old, it's hard to tell the difference between the two. And you, as parents with twins, you probably know that it's very hard to distinguish, and maybe who to punish for something that's going on, or who to bring to dinner. But as they get older, we can see that not only is one of them rocking a mustache, but they look different. You could easily distinguish between the two. One has a thinner face, a different ear shape, and in general you can distinguish between the two. And so this phenomenon had been seen throughout history, where people who had identical genomes had different phenotypes. And this was puzzling scientists for a long time, until John Gurdon in 1962 came up with this brilliant idea of taking a somatic cell, so like a skin cell, and he extracted the nucleus and put it into an unfertilized enucleated egg, and just to see what would happen. And after several attempts, he found that enucleated egg with a nucleus from a somatic cell eventually differentiated into a fully functioning multicellular frog. And so that phenomenon showed that, hey, the genes in the genome are the same in every single cell, but there's something interacting with the genome that's causing cellular differentiation and the ability for multicellular organisms to have the same genes but have expressed genes in different ways. And so this brought on the field of epigenetics. And so a different example from the Kellys is you have two mice. One has a gray and white coat, one has a brown coat. They're monozygotic twins, have the exact same DNA. But the reason that they have these different coat colors is their epigenetics, and specifically the ones I'm going to be talking, the epigenetics I'm going to be talking about today is DNA methylation. And so those are just little methylation marks on the DNA itself. And most commonly they're seen in situations where it's a cytosine followed by a guanine on the same strand. And that palindromic sequence makes it so that both strands can have the DNA methylation, and so it's conserved while cells are differentiating. But those methylation marks prevent transcription in a certain way that makes it so that different genes are expressed differently. And so that's how this mouse can have a different coat color than its monozygotic twin. And that's how, over time, these two twins can look differently, because methylation is strongly impacted by the environment, by the food we eat, the people that we talk to, and in general, not only do they look different, but their personalities are different. And this is because of their interactions with the environment. And then another more recent phenomenon found in epigenetics is the Yamanaka factors. And this was discovered in 2009, where he was able to take a cocktail of a couple of transcription factors, which are essentially proteins that also interact with how genes are expressed. And he put them into somatic cells like fibroblasts, which are skin cells. And he was able to see that those skin cells were completely reverted to a pluripotent state, very similar to how John Gurdon got to take his somatic cell and turn it into put it into a enucleated unfertilized cell, and it was able to differentiate. And so this is a type of technology that is up and coming for rejuvenating age and also rejuvenating tissues and cells. And I'll get more into that in a little bit. So Steve Horvath, he is a scientist here at UCLA, and he's exploring a similar aspect of biological aging. And he says, once we know how to accurately measure aging, we will be able to study it and to defeat it. And so this is trying to answer the question of, we know why aging. Why are we studying aging? And we just explored that. And now we're trying to explore how to prevent aging, and we believe it's through epigenetics. So why do we think DNA methylation will help us in defeating aging? And it's because, like I said, it interacts with the environments. And so it's kind of a barcode for how your cells are aging or how they're functioning over time. And then, so making a test to monitor how your DNA methylation is tracking over time will help us understand how potentially healthy you are if we can make a model that accurately predicts it. And so we've come up with this epigenetic clock idea, where if we measure the DNA methylation in a person and they have an accelerated epigenetic clock, so their predicted age from DNA methylation is higher than their chronological age, their age from the date of birth, that means that it's correlative with smoking, excessive alcohol intake, poor nutrition, stress and childhood trauma, things which are naturally associated with higher all-cause mortality. And so it's a measure of their health. And then we would also expect for that same clock, if someone was to quit smoking or to have a reasonable alcohol intake or have a balanced diet, we should be able to see a reversal of that age acceleration, and them to be having a predicted epigenetic age that is younger than their chronological age. And then there's been a lot of tests that have been developed in recent years. Most notably is the Horvath clock, or Grimm age. And then another one more recently is the Levine clock. And so the Grimm age clock, it trains its data in trying to predict chronological age. And then the Levine clock comes a little bit further and is predicting age based off of all-cause mortality score. So it's more of a test using epigenetics to determine health and health span. But ultimately, all of these clocks use blood for their sampling data. And nonetheless, the whole goal of what we're trying to do is like a genie trying to predict the future, or someone trying to predict the future, is looking at your DNA methylation and seeing how you're going to age over time. And that's the whole goal of what we're trying to do. More specifically, like I said, the limitation of a lot of those tests was through blood. And I want to give a quick analogy. So we have those of us at UCLA have all seen these vending machines around campus to take COVID tests on a weekly basis. And I just want to let you imagine if we still had to do these weekly COVID tests. Imagine having to, instead of going to one of these convenient vending machines, going to a lab and getting your blood drawn, and getting your blood drawn every week to test and see if you have COVID. And since biological age is something physicians and people should want to check on a routine basis to see if they're having a higher potential for getting certain diseases, or just checking in on how they're aging, we would want to have something more like the vending machine model, or like something like 23andMe, where you can order it from Amazon, as opposed to routinely going into the lab. And I mean, there's a lot of people that can't even go into the lab on that frequent of a basis because of anemia and other problems with needles. And so that's the whole goal, is trying to get more towards this non-invasive prediction method of biological age. So now getting to our data and how we've acquired it. So we started with this fitness study where we took 47 people who were categorized as fit, and that was through BMI and other measures of what someone would categorize as fit and healthy. And then we took 46 people who were categorized as not fit, and we also monitored their BMI, albumin, blood urea nitrogen, and about 1,000 other traits. And so this was a cross-sectional study. So we were looking at 100 people and 1,000 traits at one period of time. And then we took a buccal swab at that one period of time and did targeted bisulfite sequencing on both their blood and their buccal swab samples. And so what we're using their data for is large data analysis and making the models. And then we also have two individuals. We have only acquired their age and sex, but we have buccal swabs from them over a 3-month time period. So we're able to check and see how the models that we create from the big data set are, how variable they are with people who are taking these tests on a routine basis. Because we don't only want a test that's accurate, but we also want a test that is reliable over time. And so this is our longitudinal variation test data. And we also took targeted bisulfite sequencing of their buccal swab samples. And that's what we did for over 3 months. So what is targeted bisulfite sequencing? Really quickly, you can ask more questions later, but for the sake of time, you extract the DNA from the buccal swab, you do library preparation, pooling and concentration, and then hybridization capture. And this is the targeted aspect of targeted bisulfite sequencing. Instead of looking at the millions of methylation sites within the human genome, we're only looking at about a subset of that, which is about 100,000 sites. Or the biggest model that we were able to capture with that was high quality was about 68,500 sites. And so that saves on a lot of time and money trying to, instead of look at all of the methylation sites, we want to look at ones that we found were very useful in literature. And then bisulfite treatment, I can get into this a little bit more later, but essentially that's where unmethylated cytosines are converted into uracil and eventually into thymine. And then the methylated cytosines stay as is. And so once you eventually go into your sequencing, you're able to compare the DNA and the bisulfide treated DNA, and you're able to determine which cytosines were methylated. And then you do your data analysis, which is what we've been doing. And so our preliminary results, so we started looking at individual sites of DNA methylation and seeing how they correlated with certain traits. And starting with this one on chromosome 19, position, you can read the position. So we found that it had a very strong correlation with C-reactive protein, which is which comes out of your liver. Normally you have low levels of C-reactive protein in your blood, but your liver releases more CRP into your bloodstream if you have inflammation in your body. And high levels of CRP may mean that you have a serious health condition that causes inflammation. So seeing an increase in C-reactive protein when this position is methylated, that could be a sign of a potential problem that you'd want to get checked out. And really quickly, all of these log 10 p-values, so on a scale, or sorry, this should, yeah. So what is considered as significant traditionally in statistics is a p-value of 0.05. And in large data statistics like this, you want to see even more significant numbers, and we're actually seeing that. So log 10 of 0.05 would be -1.3. And for these top hits, we're getting about a -8, -7 log 10 p-value. And so they're extremely significant in their correlation. And then, as you can see here, for chromosome 21, at this position, it's negatively correlated with leg back strength. And this is sex corrected as well. So it's not just looking at the differences between males and females, because there is a big difference in male leg back strength versus female leg back strength. And so at this site, when it's methylated, that leads to a lower leg back strength. So you'd want to see that site as being unmethylated. And then at this site, it is negatively correlated with hemoglobin. And so we would want hemoglobin to be high, or else you could get into problems like anemia and other blood type problems or blood problems. And then at chromosome 2 at this position, it's negatively correlated with blood urea nitrogen. And this is sex corrected as well. And lower values of blood urea nitrogen could be good or could be bad, depending on the context. So that's why it's yellow here instead of red. But once we move on to looking at how they change over time, and so these are, well, violin plots and box plots of the predicted values for the individuals, which are 1 and 2, and then for the entire fitness study. And what we would want to see if we feel like these models are reliable is that the individual variation would be pretty slim and consistent. Whereas in the fitness study, we're looking at a cross-sectional study of about 100 people. So there should be a lot of variation, especially since there's variation in fitness as well, since that was part of the study. And so we should expect a large variation here and smaller variation here. Although there is less variation in individuals, this is still a lot of variation for what we're trying to see. So we moved on to a different method. Instead of looking at single sites, I've been recently moving towards this greedy algorithm. And briefly going into what that means is we use a leave one out cross-validation method to start making the models. And what leave one out cross-validation is you train the data on all of the samples except for one. And then you calculate what the test would be, so that one person, based off of the training data from all the people except that one person. And then you do that for every single sample in the data. And then that's how you build the model. And that's how we can see whether the predictions are reliable or not. I can get into that more later if you have any questions. And so how the greedy algorithm works, once we go through the leave one out cross-validation, so for all of this aspect of the algorithm goes through this leave one out cross-validation data or method. So it's done 98 times. And what it does is it filters the sites based off of how correlated they are to a trait. And so specifically the trait we were looking at here is age. And these are the top hits. This is the p-values for different sites and how they're correlated with age. And then that's sorted all the way for all 68,500 sites that we had in the fitness data. And then we do principal component analysis on all of these sites. So we take all those 68,500 sites and put them into components based off of their variation and how similar they are to each other. And then we build a linear model using those components. And then we predict age based off on that linear model. And something I didn't mention is that we do this, we can control how many sites we're using in that model. And since these are already filtered, we're choosing different amounts of sites that are highly correlated with that trait. What I mean by that is we do a log2 method where we start with 2 and then 4 and then 8 and then 16 sites. And we look at how that many sites and the models that we build from that amount of sites can predict age. And then we eventually take that number of sites and we take all of the data. Instead of doing leave one out cross-validation, now we make a model on all of the fitness data. And then we do greedy on that model. And then we predict the individual age over time based off of that linear model so that we can see how the individual people, how their predicted age changes over time. And here is an example of that longitudinal data for the individual 1 and individual 2, is we can see that at certain time points, there is large variation in their age and the predicted age that we're getting. And this is something that we want to decrease. In fact, the actual age of individual 1 is 33. And so in all of our models, we've seen that it's increased, but for us to feel more confident in the model, we want to see it more towards their actual age of 33. I mean, unless we're still figuring out whether there's a real biological component of why all the models predict that this person's age is increased, we're still working on that. But this person's actual age is 53, and so its mean is hovering right where it needs to be, but there's still a lot of variation, and we still want to make sure that we know that that variation is caused by individual, like actual traits or protein expression before we come to the conclusion that that model is 100% accurate. And then, so going back to the greedy algorithm and how we do it in a log2 method. So in this legend, we can see that we started with the top 2 sites that are most correlated with age, and then the top 4, top 8, all the way down to the maximum number of sites that we had in the data, which was 68,500. And as we can see, having all of the sites in the model actually decreased the correlation, and MAE is mean absolute error, so how much error we're seeing in the prediction. So once we go all the way up to the maximum number of sites we had, we saw a regression in how well we were predicting the age. And so we saw that at about 8,000 sites, that was the best for the correlation and also making sure that we had the least amount of error in our model. And then we also did these predictions for the individuals, and we looked at their standard deviations. So how much variation there was in their predictions over time. And so we wanted to see that being as little as possible, but obviously people do things and interact in the environment on a day-to-day basis, which could change their epigenetic age. So having some variation is still makes sense. And then again, with the mean absolute error, we wanted to make sure that that was as low as possible. And making a model off of that. So this is a scatter plot for the individuals in the fitness data set, and how their actual age versus predicted age is correlated. And as we can see, it's pretty strong. It has an R value of 0.91, which is really strong, with a log p value of -35. So it's extremely significant, and a mean absolute error of about 4, which means that on average, the prediction is about 4 years off, which is consistent with a lot of the other models that have been made, like the Horvath clock and the Levine clock, like I mentioned. And then this is a violin plot of, here's the actual age for the fitness data. So as we can see, even in the fitness data, their actual age is a little bit lower than in the fitness data than it is in the unfit data. And then for individual 1, like I said, their actual age is about 33. And the individual 2 is about 53. And we can see that individual 1, their variation is still somewhat large, but it's a lot less than the whole entire fitness data. And so this looks promising to us. And we can also see that the predictions for the fit and unfit groups are still about where they should be. The fitness groups, they're being predicted as a little bit of a younger age than the mean, and then the unfit group is being predicted at a little bit higher than the mean, just as in the actual data. And what we would expect to see if this was more of a health clock is that there would be an even starker difference between the fit data and the mean. So this should be a little bit lower, and this would be a little bit higher. So we're still looking at enhancing that aspect of the test. And so we also wanted to look at moderators. So what is making the predictions change, seeing how different traits, some of those 1,000 traits change as the predicted age is either higher or lower. And what we saw is the ones that were most correlated with either an acceleration or deceleration in age was this IgHA1 protein, which is an immunoglobulin heavy constant alpha-1 protein. That's its abbreviation as IGHA1. And it's involved in antibacterial humoral response because as a form of immunoglobulins, it is one of the versions of the constant region of antibodies, which is here. And then it's been studied recently for some forms of cancer. And as we can see, it's very strongly significant or very significantly correlated with age deceleration. So the more of it you have, then the less or the lower your predicted age is going to be, at least according to this analysis. And then S100P protein, this is a calcium binding protein, and it's involved in the regulation of a number of cellular processes such as cell cycle progression and differentiation. And it's also associated with age deceleration. And so more of this protein leads to a lower predicted age. And so you want more of it. And so systolic blood pressure was another trait that was significantly correlated or significantly moderating the age prediction, but in a negative way, as you would expect, because it's the big number of blood pressure, and high blood pressure is a serious chronic vascular disease risk factor. And so it should be associated with age acceleration, and we do see that in our model. And then angry is something that we found very interesting that actually shows up in almost all of our models as a moderator of our predicted age. It is a little bit lower. I changed this. So as a log 10 p-value of 0.01 is -2, and 0.01 is a more stringent, significant p-value, and something that a lot of data scientists are going more towards as the standard. And as we can see, angry is like right on that edge, but we still find it in all of our models, which we find significant because there's a lot of studies that have been showing that mental health is a key driver in your biological health and in general your mortality risk factors. And so we've also accumulated a lot of other data from other studies. We got data from a diabetes study and from a Turkish emergency room, and we got buccal swabs from all of about 1,000 people, including the individuals. And we wanted to see if we could build a model off of that. We only have age and sex for that amount of people, but we felt like, hey, it would be a good idea to see how, if adding this extra data would help make our models more strong. And what we saw is an increase in the R value and a decrease in the mean absolute error. And we saw that the best model was actually at about the maximum amount of sites that we had that was well represented in our quality control for across all of the samples. So we do have less methylation sites, but we showed that we have a stronger model than in just the fitness data alone. And we also see the individual standard deviation is, it's a little bit higher, but the model with about 9,000 sites is the best for this type of data, or for this data set. And so looking at that model, we can see that it looks a lot better. So back in this scatter plot, we can see that the people who are younger are predicted consistently higher than their actual age, and the people who are older are consistently predicted lower than their actual age, which might be a problem for the model. But as we can see in this model prediction, we have a lot more data, so it can train the machine learning can look at more data and make a better model. And we see that there's less of that. There's less of a skew for younger people to be predicted higher than their actual age, and less of a skew for older people being predicted lower than their actual age. So we found that to be very promising. And then especially we saw that individual 1, whose actual age was about 33, their predicted age was actually more towards their predicted age was a lot closer to their actual age. And so we found that to be promising. The variation is still a little bit high, but we still thought that was that it being closer to his actual age was more promising. And going into the moderators of this analysis of this predicted age, so I extracted the fitness data from that model and was able to run similar moderation analysis to when I was just looking at the fitness data. And we saw that C4 BPA protein which controls the classical pathway of complement activation, which is part of the immune system that enhances, complements the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism. And so it's part of that inflammation pathway. And we see that it is, again, very significantly correlated with age deceleration. So we're very happy to see that these p-values are very strong in our associations. And the next analysis would be to see how are these proteins specifically moderating age. But as of right now, these are the proteins that we find are moderating age in this model. And so A1BG, it's alpha-1b glycoprotein, and this is a plasma glycoprotein. As of right now, we don't really know its function, but it is in a lot of studies, and we can see that it's also very strongly associated with age deceleration. A2GL is another, it's a leucine-rich glycoprotein, but again, its function is a little bit unknown. And so we're just happy to see that it's negatively associated with age acceleration very significantly. And then angry, like I said, it shows up again as an even stronger association with predicted age and age acceleration. So mental health, extremely important. And so I made some plots to help look at this and how it's moderating age and its different associations. So in here, we see C4-BPA, which is associated with age deceleration. And we can see this blue line here, that's the high values. So in the fitness data set, the people that had higher values of that protein, we can see that their predicted age is consistently lower than their actual age. And then for the people with low values of that protein, it's consistently higher than the predicted age is consistently higher than their actual age. And then we can look through all of them, and that's the same case. So for the proteins, each time the higher values are associated with lower age or with lower predicted age. So it's going to be age deceleration. But then once we go to angry, it's a little bit closer here, but we can see that people who had high angry values, they were associated with higher predicted age values. And actually, so really quickly, how we determined angry, that was from a questionnaire on how frequently were you upset in the past month? Well, how frequently were you angry in the past month about things that were outside of your control? And they rated it on a scale to not often to very often. And we saw that the people that had the higher scores were the ones that had that age acceleration. And so to wrap up my talk is the idea of healthcare evolution. And so this idea of healthcare 1.0, which was yesterday, it's focused on acute and infectious diseases. And so this is like the Spanish flu, big things that we know as public health problems. And then the idea of germ theory, that germs are what are causing diseases, at least infections, as opposed to people thinking it was spiritual or something like that. And it focused more on short time frames, that a patient would come in and they would be seen by a doctor, and the interaction would probably last about a day. And the whole goal of that type of healthcare was to reduce deaths. And so we go into healthcare 2.0, which is what we are interacting with today, which is more on chronic disease management and prevention. So like cancer, we're trying to manage people with cancer and manage their illness, and also prevent it with breast cancer screenings and other types of screenings. We have big data in bioinformatics, which is what we in the Pellegrini lab are doing, and what a lot of people are doing because it's showing so much promise. And then it's focused on longer time frames. So people are inpatient for a couple of weeks or months, especially dealing with some serious chronic diseases. And preventive medicine has definitely been a forefront of what's going on, especially when the ACA came out with free preventive medicine, with checkups being free and other aspects of preventive medicine being considered more important. And then the whole goal of what we are doing right now is prolonging disability-free life, making so that people don't have morbidities for as long as possible. And then what I see as the future of healthcare, so healthcare 3.0, is an increasing focus on achieving optimal health. So instead, making sure that people are having great lifestyle and diet decisions, making sure that they feel healthy every single day, day in and day out, and having the optimal health that they can have. And then artificial intelligence, this is something that will make physicians' lives a lot easier, being able to look at maybe a systems biology approach and knowing how different drugs are going to interact and change a person's phenotype, or even going down to the genetic level, how different SNPs or how different epigenome expressions are going to change a person's phenotype, giving physicians more information to help their patients. And we're looking at a lifespan timeframe. So people are going to be going in and seeing their physician throughout their life, and they're going to consistently be going through preventive medicine. And so I say personalized preventative medicine, extreme preventative medicine, trying to prevent any illness. Like I said, trying to prevent those multimorbidities as we get older, trying to prevent the prevalence of those higher of those diseases as we age, trying to make it so that people who are in their 70s and 80s feel like they're in their 20s and 30s. And we can only really do that in a personalized way, where we're looking at each individual's genome and epigenome and using all this data for a physician to help them live as long and healthy as possible. And so I put biological age tracking and reversal. So biological age tracking, that's what we're doing non-invasively as well. And then reversal, that's a little bit into the future of using those Yamanaka factors that I mentioned earlier to partially put them back into a state where somatic cell, like your skin cell, as it ages, it starts to express proteins how it's not supposed to, but you can use Yamanaka factors in a certain way that will make it so that it's a youthful skin cell and it's actually expressing genes how it's supposed to. There's a lot of limitations with this data, with this technology right now, but I believe it can be used in sometime in maybe my lifetime, our lifetime, those of us who are a little bit younger. And so now, this guy, he is an interesting case. So he sold his company to PayPal. Yeah.
Matt, I think we need to wrap up soon. How many more slides do you have?
This is my last slide. Sorry.
Okay.
Yes.
Keep it.
So this is my last slide. Really quickly, this guy sold his company to PayPal for $800 million. And so he's really going all out on this anti-aging. And what we can see is he does a lot of different tests, like his NAD levels, heart, white blood cells, skin age, and he's doing everything he possibly can to decrease his age. And the whole goal of what we're trying to do in our lab is to make it more accessible to people, being able to get this information non-invasively and not having to spend millions of dollars. And if you're interested, I can go more into depth about what he is doing. And so acknowledgements, I have to thank Matteo Pellegrini so much for everything. He really put me under his wing as an undergrad, and I wouldn't be here without him and his help. And then Mike Thompson, he's another staff research associate in the lab that has also been a great help in acquiring this data and performing some data analysis. So thank you for listening. Yeah, I hope you enjoyed the talk, and I'm open to hear some questions.